Today we're going to look at the urinary system, and the urinary system is a little bit shorter than the last couple systems we've looked at, but we're still going to break this video up into a couple of different ones just to make it a little bit easier. The urinary system is involved with removing waste. What you'll see here is the different types of food you eat will produce different catabolic wastes. So when you eat sugar and fats, what those are going to break down to is carbon dioxide and water. How you can eliminate the waste product from those carbon dioxide is by exhaling. With proteins, you get carbon dioxide and water, but you also get these other things, urea, uric acid, and salts that are not able to be eliminated through exhaling. So we need a different process for being able to remove those. So urinary system will consist of the kidneys, ureters, urinary bladder, and the urethra. It's got three major functions. Excretion, removing waste from the body, particularly nitrogen waste. Elimination, discharging the waste. And then it's going to be involved with homeostatic regulation. So this lets you keep your fluid volume and solute concentration in the blood plasma balanced out. This diagram here is going to show the key parts of the urinary system. The urinary system has fewer organs than most of the systems we've looked at before. So you can see we've got two kidneys here, the ureters, the bladder, and the urethra. So the urinary bladder, it is a muscular sac that is for temporary storage of urine. So having the bladder in there means you don't have to constantly be leaking out a little bit all the time. The ureters are the paired tubes that are going to transport the filtrate, which is your urine, to the bladder. The urethra is going to be the tube that takes it outside the body. And micturition is the process of urination. So you do contract the muscular urinary bladder to help force out the last little bit in there through the urethra. This is a transverse section of the abdomen, and this allows you to see where the kidneys are located. They are located back behind the abdominal cavity here where you have most of the organs. So for the kidney location, we actually say that it's retroperitoneal because it's behind the peritoneal membrane. Occasionally you will run into renal ptosis, which is a floating kidney. This is a condition where the kidney can basically descend more than 2 vertebral bodies, which is a significant size. It's over 5 centimeters when you go from being supine to upright. So when you first find this on a patient, it will seem kind of odd to feel it. It can lead to other problems where sometimes in floating around, it can float into places you don't really want it. So it's not an ideal situation. The nephrons, these are the functional units of the kidney. You're going to have between 1 to 1.5 million nephrons in there, and they are two main types. These are going to be your actual filtering organs. So this is a nice diagram here of the kidneys that gives you the main structures of the kidneys that lets you see it. It's not overly detailed. And then it's going to show you the path that the urine is going to go through after it comes out of a nephron right here, goes into a collecting duct, papillary duct in the renal pyramids, the minor, and then the major calyces, the renal pelvis, ureter, and urinary bladder. So it's a nice summary in that picture. For our two types of nephrons, we've got cortical and juxtamedullary. The cortical ones are responsible for removing waste. They're going to do the production and reabsorption of the nutrients, where the juxtamedullary are more involved with concentrating the urine. So concentrating the urine is important because you would not want to have to take in all of that fluid that you've lost. The renal hilum is the indent in the kidney. This is where you're going to have the arteries, veins, and nerves come in and out, as well as the ureter. So if we go back up here, this is going to be your renal hilum, the little indent. When we look at the external anatomy of the kidney, it's got 3 layers of tissue. The renal capsule is going to be the deeper of the external layers. and it's going to be continuous with the outer coat of the ureter. This provides a fibrous barrier that's going to protect against trauma and help maintain the shape. The adipose capsule is going to protect it from trauma as well. It's basically fatty cushioning, helps hold it in place. And then the renal fascia is the superficial layer. It's thin connective tissue that helps to anchor it in place to the surrounding structures and the abdominal wall. So the inner anatomy, you've got the renal cortex. This is the most superficial of the inner anatomy. You've got the outer cortex zone and inner juxtamedullary zone. The renal medulla is the inner region, so it's the whole inner region. The parenchyma is the functional portion. This is going to be where you've got the renal cortex and renal pyramids of the medulla. So when we look at the excretion of any solute, it's going to be your glomerular filtration, any secretions minus what's reabsorbed. So here you can see this is the cortex, and then this is the medulla, the inner portion of the kidney. So the renal pyramids are going to be where the nephrons are located, and the space between the renal pyramids is the renal column. So that can be seen on this picture as well. The darker pink is the renal pyramid. The renal column is the lighter pink in between it. The calyces are in the periphery of the renal pelvis. So they're around the outside. They're little cup-like projections around the outside. The minor calyces are going to surround the renal papillae of the papyramid. So down at the bottom is where you've got the renal papillae. They're going to act like a cup to collect urine from the pyramid. The minor calyces will come together to form the major calyces. So urine will flow from the major calyces into the renal pelvis and then into the ureters to exit. So looking at the nephron, there's over 1 million of them. So the renal corpuscle is the site of the plasma filtration. It's going to be made up of your glomerulus and the glomerular or Bowman's capsule. The glomerulus is a capillary bed where the filtration occurs, and the capsule is a double-walled epithelial cup that's going to surround it and collect the filtrate. Then it will move into the renal tubule that's got 3 parts: the proximal convoluted tubule, the loop of Henle that's going to dip down into the medulla, and the distal convoluted tubule. We'll get more into what happens in those areas a little bit later on. And then it will go into the collecting ducts and papillary ducts that will go into the renal pelvis and ureter. This is a nice picture here of a nephron that I like because it does give some nice little explanations, some arrows showing what's going on here. That's a simplified picture. That you can see here you've got your glomerulus, the Bowman's capsule, proximal convoluted tubule, loop of Henle, distal convoluted tubule, and collecting duct. So this is going to show an enlargement here of the renal corpuscle that's going to have your Bowman's capsule or glomerular capsule. And then you've got your glomerulus inside. You can see that it's a capillary bed, and the fluid will filter through those walls and get caught into this cup and then move into the proximal convoluted tubule. So you've got podocytes that cover the capillaries to form the visceral layer, and then there's simple squamous cells that are going to form the parietal layer. So along the way, you are going to have peritubular capillaries. So these peritubular capillaries that you're going to see in some of the pictures later on are important because it's going to help in returning blood back to the venous system. So much is filtered through here. If we don't recapture most of that blood and fluid, it would be an overwhelming task for the body to have to replace all of that. So that renal tubule is a long tubular passage that is going to work to help reabsorb useful organic nutrients, water, and then allow the waste products to go out. So those three parts: proximal convoluted tubule, loop of Henle, and distal convoluted tubule. So here is a picture that's a little bit more realistic of a nephron, where you've got all of these blood vessels wrapping around here, peritubular capillaries wrapping around to help pull things back in. It's also much more twisted up. That picture up top, the nice thing is it's been untangled to see, but reality is they're very twisted up in the kidneys and very dense in there. So each nephron empties into the collecting system, which is a series of collecting ducts that will carry the fluid away from the nephron. The number of nephrons you have remains constant from birth, so you don't replace them. However, we do have a lot of them. Kidney dysfunction is not actually going to be evident till it declines to about 25% of normal. So what you have from birth is actually a lot more than what you need. However, you're never going to replace it, so you don't want to damage it. But it can take quite a bit of damage before it's going to be noticeable to the person. So if one of the kidneys is removed, the other one will actually enlarge a little bit till it can filter at about 80% of the normal rate of 2 kidneys. So it can expand its function a little bit in there as well. So the kidneys are able to clear the waste products from the blood. This is important to maintain your water and electrolyte balance. When we look at the blood flow, the renal artery is going to supply blood to the kidney and the renal vein is going to carry it away. So that's the larger vessel. Human kidneys are going to receive about 25% of the blood pumped through the heart. So they're large vessels that go there. The arcuate arteries and veins are going to be part of the renal circulation, and they're at the border of the renal cortex and renal medulla. And then you've got the cortical radiate arteries that are going to radiate out from those arcuate arteries and branch into numerous afferent arterioles. Those are going to be the ones that are going to enter the capillaries supplying the nephrons. So the order of blood flow, it will go from the capillaries to afferent arterioles to the glomerulus, then back through the efferent arteriole. Some of it will return through peritubular capillaries and the vasa recta to the cortex and medulla. So this picture down here shows where you've got all of these little blood vessels that are around the outside here, where you would have them go into the nephrons. So the afferent arteriole is a small branch of the renal artery that is going to go to the glomerulus. The glomerulus is that little capillary bed in the Bowman's capsule where you start the filtration. The efferent arteriole is going to be a small artery branch that's going to go away from the glomerulus. So you're going to have blood filtered initially in the glomerulus, and then it will leave there. What's been removed from the glomerulus is going to be your glomerular filtrate that will get processed later on. Peritubular capillaries are going to go around the renal tubules. So that way, when you have that filtrate go in there, this is going to allow for immediate reabsorption of some of the essential substances from the fluid. And this can happen in the proximal convoluted tubule. The vasa recta is the capillary network around the loop of Henle. So you got more major exchanges of water and salt taking place here so that you could try and conserve things. The proximal convoluted tubule is the one nearest to the glomerulus. You'll start reabsorbing some of those essential substances. You've got simple cuboidal cells with the brush border of microvilli to help increase the surface area. So the U-shape part of that tube is the loop of Henle, and it will have a thin descending limb and a thick ascending limb. The ascending loop is going to have simple cuboidal to low columnar cells, where the descending loop is simple squamous cells. And we're going to look at that in more detail as well. Your distal convoluted tubule, this is going to be between the ascending loop of Henle and the collecting duct. Here you'll do the final concentration of the urine filtrate, or at least start the process. You'll have simple cuboidal cells And then you have principal and intercalated cells that are going to have microvilli on them. So here again is another picture of a nephron taken out of a kidney. And you can see you've got your loop of Henle here that is going to dip down. Your renal plasma flow is the volume of blood that passes through the kidneys per minute. This will be 600 to 700 milliliters. So just like we measure how much passes through the heart in a minute, we measure how much passes through the kidneys. So between each kidney, at being 600 to 700 milliliters apiece, your total renal blood flow is about 1,200 milliliters per minute. So again, this is kind of a nice picture here that it shows the blood flow over to the right. It pulls out a nephron so you can see what happens, and then where the nephron would be located in the pyramids. So glomerular filtration, you've got this filtration barrier that's made up of endothelial cells of the glomerular capillaries and podocytes that encircle the capillaries. So it forms a coil of about 8 capillary lobes in there, and it's going to form a non-selective filter of plasma substances that have a molecular weight less than 70,000 Daltons. So it's pretty small stuff. It's going to permit filtration of the water and the small solutes, and it prevents filtration of the plasma proteins via the basal lamina, the blood cells, and the platelets through the fenestrations and pores of the glomerular endothelial cells. So it's going to keep in some of the bigger stuff. Here you can see this is an example of where you would have the capillary bed in a glomerulus under the microscope. So it's the net filtration pressure which forms your total pressure promoting filtration. So we have an equation for this. The NFP equals the GBHP minus CHP minus BCOP. So it's your net filtration pressure is going to be equal to your glomerular blood hydrostatic pressure minus the capsular hydrostatic pressure plus the blood colloid osmotic pressure. So in here, this isn't really all minus, it's just these are the components of it. So your glomerular blood hydrostatic pressure is the blood pressure of the capillaries. So how much pressure is in those capillaries that's going to push the water and solute through those little slits that cause the filtration. Your capsular hydrostatic pressure is the pressure exerted against the filtration membrane by fluid that's already in the capsular space. So this acts as your back pressure. And then the blood colloid osmotic pressure is due to the presence of the proteins in the blood of the plasma. So this also opposes filtration. So things that influence this filtration process: the cellular structure of the capillary walls and the Bowman capsule. So this filtrate has to pass through the 3 glomerular filtration barrier layers. It's got to go through the capillary wall membrane, the basement membrane or the basal lamina, and the visceral epithelium of the Bowman's capsule. So the capillary wall endothelial cells are going to have certain pores, we call them fenestrated, to increase the permeability, but it still is not going to allow the large molecules to pass. Those large molecules are still further restricted by the basement membrane podocytes that form these intertwining foot processes. So the cortical nephrons, these are 80 to 85% of the nephrons. They're in the renal corpuscle, in the outer portion of the cortex and short loops. They only extend into the outer medulla. They don't go as far down. So here you can see it's in the outer portion of it. It's only part of the loop of Henle going into the renal pyramids. The juxtamedullary nephrons are the other 20 to 25%. They're going to secrete very dilute or very concentrated urine. So they're deep in the cortex. Their long loop of Henle is going to go deep into the medulla. They're going to get blood from the peritubular capillaries and vasa recta. And the ascending limb is going to have both thick and thin regions. So we have a shield of negativity. In addition to the structure of the glomerular barrier, this is going to repel molecules with a positive charge, even if they're small enough. So that charge will influence what gets through as well. This is going to help keep the albumin, which could actually pass, from going through. You want to hang on to the albumin. So the glomerular pressure, this is hydrostatic pressure that is going to be the result of the small size of the efferent arterioles and the glomerular capillaries. That's going to help to enhance filtration. It's basically the pressure exerted by a liquid. The osmotic pressure is going to be the pressure of the substance in the solution due to the presence of colloids. And then when you increase or decrease the size of the afferent and efferent arterioles, this can happen with an autoregulatory mechanism within the juxtaglomerular apparatus. It helps to keep the blood pressure in there at a constant rate. This will happen regardless of the fluctuation in the systemic blood pressure. So the pressure of blood is very important for the filtration of kidneys. That's one of the reasons why kidneys are so susceptible to damage from diabetes, is the blood sugar in there will damage those little tiny blood vessels and affect the pressure. Kidney disease is one of the organic causes of hypertension. So they are closely tied in with the blood pressure system of the cardiovascular system. So if you have a dilated afferent and constricted efferent, this would happen when blood pressure is too low. So this would create a decrease in blood flow to the kidneys. If you have a constricted afferent and dilated efferent, this happens when blood pressure is too high. This prevents overfiltration and damage to the glomerulus. So then the renin-angiotensin-aldosterone system is also going to have an influence here. This is going to regulate the blood flow to and within the glomerulus. This will be affected by changes in blood pressure and sodium that's monitored by the juxtaglomerular apparatus. So that's going to have juxtaglomerular cells in the afferent arteriole and the macula densa of the distal convoluted tubule. So when the plasma sodium's low, it decreases water retention. That will decrease the blood pressure and blood volume. So when the macula densa senses this, it's going to trigger the renin-angiotensin-aldosterone system to raise blood pressure back up. The renin-angiotensin-aldosterone system is also a place where pharmaceutically blood pressure gets controlled. So what happens is renin is going to be produced by those juxta cells that react with angiotensinogen. It will form the hormone angiotensin 1. That goes through the alveoli of the lungs, and the angiotensin converting enzyme will change it to angiotensin 2. Angiotensin 2 corrects renal blood flow by either doing vasodilation, stimulating sodium to be reabsorbed and water in the proximal convoluted tubule, or triggering the release of aldosterone from the adrenal cortex. Aldosterone is also known as an antidiuretic. It will cause the body to retain sodium and water and increase the blood pressure and blood volume. So the lung health can actually affect this system as well. So the kidneys are definitely a place where you can see how different systems of the body are very intertwined. So when systemic blood pressure and plasma sodium rise, renin is going to decrease. So for tubular reabsorption, the nephrons need to reabsorb 99% of the filtrate back. Since the glomeruli filter about 120 milliliters of water per minute with low molecular weight substances, you don't want to lose all of that in a minute. The nephrons need to reabsorb most of it. The process is both active and passive. There's osmosis involved for water reabsorption, pinocytosis for small protein reabsorption, and then cellular mechanisms that are also going to be involved. So we have some active transport that will be involved here. In order for this to happen, the substance has to combine with a carrier protein in the membrane of the renal tubular epithelial cells, and then it will use electrochemical energy to transfer the substance and put it back into the bloodstream. So things we would use active transport for: glucose, amino acids, some of the salt in the proximal convoluted tubule, chloride in the loop of Henle, and sodium in the distal convoluted tubule. With passive transport, you're not going to be using energy here. You've got movement of the molecules across the membrane that is the result of a difference in the concentration or electrical potential on opposite sides of the membrane. So it's going to move down gradients. This takes place in all the parts of the nephron except the ascending loop of Henle, where the walls are impermeable to water. So urea is passively reabsorbed into the proximal convoluted tubule and the ascending loop of Henle. Sodium will passively be reabsorbed into the ascending loop. So your maximal reabsorptive capacity, or transport maximum, this is when the concentration of filtrate exceeds this. So if you have too much in the filtrate, it's going to start to appear in the urine. It can only handle so much. The renal threshold is the plasma concentration where active transport stops. So for glucose, if it's over 160 to 180 milligrams per deciliter, you're going to start to see glucose be in the urine. So diabetes is usually diagnosed at about 150 milligrams per deciliter. So one of the ways that they would see in the past if a person was diabetic is there would start to be sugar spilling into the urine. And so before modern blood tests or urine tests, what they had to do was have somebody taste the urine. So then we have reabsorption in the proximal convoluted tubule. This would include glucose, amino acids, lactic acid, water-soluble vitamins, and other nutrients that are completely reabsorbed in the first half. So this picture here shows you've got your fluid in the tubule of the lumen. This is showing some active transport here. It's using ATP to move the glucose out, and then the glucose can go back into the bloodstream. So this is showing it here in the proximal convoluted tubule. So this is in the second half of the proximal convoluted tubule. This is showing here where you've got these ions moving by diffusion and then water moving by osmosis. So tubular concentration. The renal concentration begins in the descending and ascending loops of Henle. This is because of the high osmotic gradient in the renal medulla. Water is going to be removed by osmosis in the loop of Henle, and then sodium and chloride are absorbed in the ascending loop. So why does all of this matter? If you know what components are in the urine and when you have excesses or some missing, this can help you determine what part of the kidney or urinary system is not functioning properly. So that's why it's important to know where in the kidney the different things are being absorbed. So when we look at the reabsorption in the loop of Henle, the descending limb is highly permeable to water. So you have reabsorption that can occur passively. You've got low amounts of urea, sodium, and other ions absorbed. In the thin part of the ascending limb, this is impermeable to water. It doesn't have any aquaporin channels. Sodium reabsorption still occurs passively through the epithelial cells. And then you have chloride ions that are reabsorbed into the thin limb through chloride channels. And then some paracellular movement of the sodium and chloride due to the differences in the osmolarity. And then in the thick ascending lumen, this is the primary site of sodium reabsorption. It's impermeable to water. Sodium reabsorption is going to be active. It's going to be drove by the enzyme of the sodium-potassium ATPase on the basolateral membrane. It's going to pump 3 sodium ions out and 2 potassium ions in. So this will create a low intracellular concentration of sodium. The inside of the cell will become negatively charged and create an electrochemical gradient. So this is a diagram here that's going to show, again, your sodium and potassium pump, moving the sodium this direction, the potassium and the chloride, and then the potassium being returned back, and things being able to move back into the bloodstream. So the countercurrent mechanism, this is going to help to maintain the osmotic gradient of the medulla. This is going to keep it from having too much water reabsorption through the impermeable walls. The filtrate leaving the ascending loop is going to have a low concentration because of the reabsorption of sodium and not water. And in the distal convoluted tubule, sodium reabsorption is going to be under the control of the hormone aldosterone. So here you can see what's moving through. You can see the different concentrations of the filtrate as it moves through here. So we will stop and pick up in the next video.